and he works with the Field Epidemiological Laboratory Training Program, Kenya. So welcome so much, Duncan. I encourage all of us, if you have any question, you can always post them in the Q&A session. I mean the Q&A box, so that after the end of the presentation, we may go through them. Otherwise, Duncan, are you able to hear me?
Yes, Bruce, I can hear you.
Thank you so much. Welcome, and I'll be moderating you. My name is Bruce Onyango. Welcome, Duncan.
Thank you so much, Bruce. Let me take this opportunity to say good afternoon, everyone. Those who've gotten time, though despite we are morning, those who've gotten time to join this wonderful webinar today. So I'll also take the opportunity to thank Laboratory Society of Kenya, Laboratory Medicine Society of Kenya, for giving me the opportunity again today to share with my colleagues this wonderful topic that we want to discuss today. So today, I'd like to take you through what has already been introduced by Bruce. That is strengthening a laboratory system to help us in early detection and monitoring of disease outbreaks. So Fred, kindly confirm if you can see my screen and if I'm audible enough.
Yes, Duncan, you are very much audible. We can see a screen, though it's not full. We can see half of it.
It's not in, let me put it in presentation mode. What about now?
It's still half. Of course, it's in the presentation mode, but it's half.
It's half.
I don't know whether, we don't know whether it's the layout.
I think it should be fine.
Not really, not still fine.
And it is.
But the participants can react.
Okay, can you try minimizing it? You can minimize the screen if it's possible.
So let me share again.
No. It's still not full.
It's not full yet.
Not full yet.
What about now?
Okay. Yeah, it's fine.
It's fine. So thank you so much for that. Sorry for that technical itch. So I'd like to take you through laboratories strengthening for surveillance and outbreak monitoring and early detection. So the topic for today, as we all know, that there's no outbreak that can be can be confirmed in Kenya without laboratory confirmation. So laboratory plays a very important and very pivotal role in outbreak and surveillance in Kenya. So we are very, very much important. And I'll take you step by step just to take you through why the laboratory bit of it is very much important. So maybe just an examples, just to give you an examples of what has actually been, transpired in the past. During COVID, the cabinet secretary could not announce or maybe declare the outbreak of COVID in Kenya without laboratory confirmation. So despite having all the clinical and even those clinical and everything, you cannot declare an outbreak without laboratory confirmation. So laboratory is very, very, very key in early detection of outbreaks in Kenya. So what are some of the outbreaks that we've had in Kenya in the past? So we've got COVID, and lately the country is also having some other diseases like cholera, which is very much important. And laboratory plays a very pivotal role in detection and confirmation of cholera cases, and and uh, and maybe declaring the end of a disease. So, like just to give an example, how what is the importance of laboratory in cholera confirmation and detection? So, as laboratory, we are expected uh, from the symptoms, that is, we've got the symptoms, we've got suspected symptoms, we've got the probable, and we've got the confirmation. So if you brought the sample as the laboratory, you are supposed to do two tests. We've got now we've got the RDT, the rapid test that are usually done. Then you've got to confirm it with a culture test. So in this case, we are supposed to confirm cholera using what we call culture. So this culture, you can also go ahead and do some genomics on it. So just to get the kind of strains that are required. So from there, then it can actually be declared. But alone using the same signs and the symptoms of cholera, you cannot actually declare cholera or any other disease. Maybe to talk about maybe the mpox and the rest. The clinical signs are there, but what is now the confirmation bit of it, which is very much important. So I'll walk you through so that we can actually appreciate ourselves as laboratory officers, me included, that laboratory plays a very pivotal role. And how can we enhance these systems so that we can actually pick it? When we basically talk about malaria, when we talk about cholera, when we talk about hepatitis, when we talk about these Rift Valley fevers, we've got so many diseases which are in the radar of the WHO. When we talk about cultures, how can we strengthen laboratory systems so that we can do the early detection, and we can also, the bit of it is now the how can we advise the clinicians on the collection that is pre-analytical, analytical, and post-analytical, which is now basically the introduction, which is basically the interpretation of the results that we generate from the laboratory. So I'll be taking you through that today. That one was just a little bit of introduction. So what is an outbreak? So it refers to the process of enhancing the capacity, capability, and quality laboratory systems to detect, confirm, and monitor infectious diseases at the earliest possible stage. So the key word or take home message is earliest possible stage. Are we able to detect when we talk about cholera? I want to give an example like in our settings. We've got what we call IDSR reporting tool. IDSR is basically, it means integrated disease surveillance and response that we do usually fill in our laboratories. So we collect the data, and the data is analyzed by the public health or the surveillance team. So it usually collects the malaria, which is done on weekly basis. It collects what we call any diarrheal diseases. Those are the dysentery. Then we've got the salmonella, that is now the typhoid cases. Then any other. It's usually picked through what we call IDSR 545, MOH 545, of which I think we are very much familiar with this. So you are called upon as laboratory officers. These malaria tests that you usually report on weekly basis, have we monitored the trends? Do we have any upsurges? The typhoid cases that because we know salmonella generally is a very is one of the bioterrorist organisms. and it should be handled most of the time with care, because it can actually bring a lot of issues. So just one case of salmonella accounts for an outbreak. Only one case of mpox accounts for a case. Only one case of bacillus or anthrax case calls for a case. So we should also know the thresholds. Do we know as a facilities or as laboratory officers, because we are the people who are doing this? Do we really know that? Then number two, it includes improving infrastructure, equipment, personnel, procedures that are talked about, that we've got to know about pre-analytical, analytical, and post-analytical. As the saying goes, that garbage in, garbage out. So whatever you sow is what you will reap. So if you work well in the pre-analytical phase, then most probably you would actually get a proper, if you want to get a proper result and correct and quality results, then you've got to work on the pre-analytical phase so that you get the end product becomes the best. Then what about laboratory surveillance? So surveillance is generally the collection of which we are doing the laboratory. We've got all the MOH tools and reporting tools that we've got in our facilities. Just to talk about maybe the tools that we've got in the lab, we've got what we call MOH 240, and we're also failing MOH 545, which is the disease surveillance, which deals with that. Then on that, we've also got several registers in our facilities, maybe just to talk about, like we've got also MOH 706, which is the laboratory summary, a monthly summary register. We've got MOH 743 and the rest. So we should actually be conversant with this, because the data that we really generate in our laboratory is very much important, and we need to analyze this data so that we come up with the trends and we monitor the trends for any early detection of any disease, any outbreak. We need to monitor the trends. We come up with the trends. This one can be actually be done through graphs, can be done through line graphs, can be done through histograms, can be done through pie charts, and also you can do some little bit of descriptive analysis. In our last lecture, I talked about the descriptive analysis. It was, you've got to describe the graphs that you come up with in terms of time, place, and person. That is TPP, which is very much important. What are you basically talking about? Is it malaria? Which group is it affecting? And what is the time of that occurrence? So that we get to monitor some kind of seasonality. So it also involves tracking of confirmed cases of disease through laboratory diagnosis, rather than relying solely on clinical reports. So underline the word clinical reports. So that's why I said in my opening remark that laboratory plays a pivotal role, almost 99.5% in outbreak in response and outbreak, generally in outbreak response. So you can't rely on the clinical reports, because clinical reports, we've got the suspected case, you've got a probable case, and a confirmed case. And the confirmed case comes at the tail bit of it, which is now the confirmation. And this one relies on the laboratory, whether it is through rapid diagnostic tests or through culture or through PCR diagnosis or diagnostics, genomic sequencing. Very much important. It also involves laboratory surveillance provides objective confirmatory data that strengthens the accuracy, reliability of disease surveillance systems, which is very much important. The word accuracy and reliability, so that once you come up with a test, you've done a test, it should be accurate and it should be reliable. That once this is taken or referred to another level, the same results will be gotten from that, so that we avoid issues of trust. You do a quality test, we produce quality results. So a little bit of the background again. Surveillance reports provide baseline data about the normal disease occurrence in a place. Then when laboratory surveillance data shows cases exceeding baseline levels, outbreak detection triggers investigation. So I would actually want to pose this question to my fellow laboratory officers who are on board, that how many times have we monitored? Do we really know even the threshold of our malaria? Because the threshold is usually divided into two. We've got the alert threshold and the action threshold. So the action threshold actually tells you that you are supposed to act. Do we really know those ones? Because the thresholds are usually calculated from a facility data over a period of 5 years, of which one that is very much important. Then alert threshold, it just alerts you that there's something that is there's something that is wrong actually somewhere, and action, you are supposed to act on it. So we've always had the alert thresholds. When we basically talk about malaria, there's alert threshold for malaria, and we've also got action threshold for malaria. When we talk about hepatitis, there's an alert and action threshold. When we talk about cholera, there's alert and action threshold. When we talk about Rift Valley fevers, maybe for our fellows that are in arid and semi-arid areas, that is Isiolo and the rest, the northeastern period, part of Kenya, there's this disease that is usually common. It is usually zoonotic. It affects both human and animals. And in animals, it usually presents with abortion cases in animals, while in human, it usually presents with fever. So in that case, it's usually called Rift Valley fever. So we should actually know if a clinician comes to you and would actually want some kind of help, like advisory services, ask you, how can I collect this sample, and how can this one be diagnosed in the laboratory? So you know how you can actually advise. So the advisory part of it, when it comes to outbreak investigation and detection, lies solely with the laboratory. How do we advise our clinicians and the clinical bit of it on the sample collection? How do we collect all of this and this and this and this, so that we get a quality outcome at the end of it all? Then there's integrated approach that ensures laboratories are not just providing test results, but actively contributing to rapid disease detection and control response. So I think the point is on that we are supposed to provide test results, then we actively contribute to rapid detection, which we are doing, and we control the response. So this one is just like in cholera. After you've detected cholera, you've done the rapid test, and the is positive for cholera. You've done the culture, and the culture is positive, and maybe you went ahead and did maybe the PCR, the genomic sequencing, and maybe you've isolated the Inaba or Ogawa cases. You're supposed to advise. So advising on the response, how would one know that the cholera cases have subsided? So it depends with the samples that you are receiving in a laboratory. If you get the 10 cases for the last, maybe for since you started, and the cases are now subsiding, you're supposed to use what we call epidemiological curve. So the epidemiological curves is usually just what is usually plotted in the laboratory. You can plot it in the laboratory, or you can just plot it. It's simply the number of cases that you've got and the dates. So in this date, I actually received like 30 cases, and they were positive. On this case, I got this. So in this case, I got this. So if you plot that, I think this one I took you through, those who attended my lectures last time, on how a graph can actually be plotted. So if you plot that graph, you will actually know when were the peaks, are the cases subsiding, or are the cases are on the verge. So what is now the key components The key input components we've got is divided into 5 categories. I talked about the IDSR. The IDSR is Integrated Disease Surveillance and Response, and this one is MOH 545. And it talks about priority diseases by health threat level. So we've got category 1. So these are the most urgent and should be reported immediately. We've got cases of yellow fever, polio cases. We've got acute flaccid paralysis, meningitis, Marburg, Ebola virus, cholera, influenza with severe respiratory infection. If you come across any of these cases, like maybe you've done a culture of the CSF, or you've done the serum for meningococcal disease, you need to you need to alert, or you alert the higher officers immediately on that. Then secondly is high priority. We've got the measles. I think measles, majority of the cases, most of the cases, we usually collect the serum, then we send them to we send the measles samples to maybe to national public health labs for analysis, the same as pertussis and tetanus, a typhoid fever and dysentery, maybe if at all we've got the cases. But the underlying thing in this is that we've got to know the kind of samples to be collected and how this is supposed to be packed, whether we need the triple packaging, or how can you package this? What are the temperatures for this? What are the temperatures? And what is the culture media that you need maybe for the transportation while after you've collected the samples? So those ones we need to put in place, and we need to advise the authorities on the same, so that by doing this, we are geared towards quality results at the end of it. Then we've got TB under category 3. We are very much familiar with TB, MDR, and the rest. We've got the sleeping, we've got the plague, and brucellosis. Then category 4, we've got malaria, we've got dengue fever, which is very important. We've got whooping cough and meningitis. Then acute under 5, acute respiratory infections, that is acute diarrheal diseases and acute jaundice syndrome. So basically that one was just under the priority of diseases. So majority of the diseases that I've talked about, majority of them we handle as the laboratory. The question would be, how are we managing this? Are we communicating according to the category? Because the standard detects that according to the category, that the ones that if you get a case, just one case, you're supposed to immediately report it, so that an action can actually be taken. A response team after you've reported it should be initiated, so that it be investigated. Because failure to do that then is going to affect a large kind of population. So these are the diseases targeted for weekly reporting. I talked about that: cholera, typhoid, dysentery, we do, or measles if there is a case, or meningococcal, we've got a plague, yellow fever, viral hemorrhagic, and neonatal tetanus. And lastly is malaria. Very much important, because it's us in the laboratory that we diagnose the malaria in terms of the counts, that is speciation vis-a-vis the quantification. We are able to do that. Detection, speciation, and quantification, which is very much important to us when it comes to malaria. We've got to know the thresholds that are talked about, the alert and the action thresholds, which are very much important. In terms of dysentery, after the culture, are we able to do the culture and sensitivity? What are some of the organisms that we come up with after that, so that we can actually conclude that this one was this kind of organism. Is it organism of medical importance? Can we report that? Then cases of yellow fever and the rest. So about the disease thresholds that I talked about, what is the alert threshold of a cholera case? So alert threshold of a cholera is just a single case of a cholera case is suspected, and that one is alert. And in terms of action, when do you act? If a suspected case is confirmed, if you've got to act when a suspected case is confirmed. What about typhoid fever? Alert threshold is observed when there's an increased number of cases or deaths over a period of time. So a period of time could even be maybe 1 week, 2 weeks, or even a month. So we've got the underlying point is you've got to monitor, and we've got to monitor all the cases that we generate and all the data that we generate in the laboratory. What about dysentery or diarrhea with blood? The alert threshold is observed increase in number of cases or deaths in a period of time only when you are monitoring this in the laboratory. When do you act? You act when observed above with at least one case confirmed through culture and sensitivity that I talked about. If you confirm it, something that is done, then you confirm it through culture and sensitivity, then you've got to act. And in this case, we've got to alert our public health. We work in collaboration. We inform them, then we act on the same. What about malaria? The alert threshold is usually attained when a number of weekly malaria cases observed crosses above the third quartile level of the 5 years. I talked about this one in malaria is usually a periodic bit of it that you've got to monitor over 5 years, and you're supposed to come up with what we call the standard deviation, mean and the standard deviation of the malaria cases within 5 years, a period of 5 years. So alert thresholds indicates there's a problem, and there's what we call malaria upsurges. So this one cannot usually be monitored. Nowadays is digitized. We just go for those of us who've got KHIS, that is KHIS rights, Kenya Health Information Science, that is DHIS2 rights. You are able to monitor the alert and the action thresholds of malaria through KHIS, and you are able to do that effectively. In terms of TB, the alert threshold is usually observed increase in a number of cases or deaths over a period of time. When do you act? When it goes two folds, it twice what has always been in the cases in the past. So we also got VHF, that is viral hemorrhagic fevers. So viral hemorrhagic fever capsules, it has a lot of cases, like we've got Ebola, we've got Marburg, so many cases. So in alert, just a single case accounts for an outbreak. When do you act? When the case is confirmed. So the case is confirmed through laboratory confirmed case. Then you are able to act. What about in yellow fever? Just a single case, the way I had mentioned, calls for an outbreak. A plague, also a single case also. If it's confirmed through laboratory, the same. The same as, what about in pneumonia? Observed case increase in number of deaths or over a period of time. Action, if it's twofold and there's a death that has already been reported. So what is the outbreak significance? Apart from the test that you talked about, we've got now the rapid test, and culture and the genomic sequencing. What is the significance? So they help us in the case identification and confirmation. So laboratory continue testing suspected cases to identify all confirmed cases. Measles outbreak, all suspected cases usually undergo testing. Laboratory testing and confirmed cases are recorded with demographic and clinical information. So why is demographic and clinical information very important? I talked about time, place, and person when you want to plot all the cases that you generate in the laboratory. So time will tell you how many cases were reported at this particular time. Then place, what is place? You can actually use what we call the plotting graph. You can use a graph. For those of us who know how to plot the graph using what we call QGIS, which is basically maybe an app of just generating the maps, it will show you which region is most affected by this disease. That's why having the demographic and the clinical information, then the clinical information would actually tell you in terms of sex, that is gender, and in terms of age group, which age group and which kind of sex is most affected, so that this one will inform when you want to do what we call intervention. So when you want to have what we call targeted intervention, you go for this demographic and clinical information, which is very important. Just like the register that we've got in our laboratories, or those of us maybe who was using, we usually have those kind of variables. We call them variables. So age, sex, type of test, residence, the outcome of the test, and the test that has actually been done. Very much important. The period, the month, date of the test is done, age, sex, and the rest. Why do we need those ones? They are not just placed there just because they're supposed to be there. There's a reason why we've got those kind of variables in our registers. So age shows this, sex shows this. So we can analyze all those variables and come up with what we call the trend analysis in our laboratories, which is very much important. It also helps us to come up with the trend analysis. It also helps us in epidemiological investigation. Laboratories provide isolates of strain typing or molecular characterization that helps understand transmission patterns. So by plotting graphs, epidemiological graphs. So epidemiological graphs, what are epidemiological graphs? So our registers, or what we always use in MOH 545, that is IDSR, it has what we call epidemiological week. So epidemiological week runs from Monday to Sunday. That is epidemiological week 1. So that is Epi week 1, then until Epi week 52nd. So week 1 starts from 1st of January, 7 days, like that, until December, the last date. So it has what we call epidemiological weeks. It totals to 52 epidemiological weeks. So if you are to plot them, you can actually monitor the trends within the epidemiological weeks, which is very much important. This one will also tell you about the seasonality when you want to talk about maybe malaria. So which season recorded this and which one this one had this, so that you monitor the trends over time for target interventions. Okay. Then evaluations and interventions. This one is just monitoring. Like we've got drug efficacy research that are usually done, maybe in terms of vaccination. You would want to monitor interventions. Are the drugs working or not? Like when you want to talk about malaria test and you want to monitor the resistance of AL in our laboratories. So we collect a sample using the DBS dry blood spots and is analyzed using PCR, polymerase chain reaction. We analyze it for what we call resistance markers. So these resistance markers are usually called the K markers. That is K, to be precise, is K13 for malaria interventions. So early detection and outbreak investigation. Why early detection and outbreak investigation in our laboratories? So it strengthens our lab to enable rapid identification of the disease outbreaks. I talked about that. Unusual cluster of respiratory illnesses usually occur in a community. A well-equipped laboratory can quickly conduct a molecular test to identify whether it's influenza, COVID-19, or another pathogen, enabling swift public health interventions. Then in foodborne illnesses, outbreaks, labs can identify bacterial pathogens like Salmonella species within hours rather than days. So that is now the swiftness that I talked about, so that we work on the time. Then effective surveillance requires rapid data flow from laboratories to public health authorities. This one I find is very much important about the transport media. As a laboratory officer, a clinician comes to you and would want to ship a measles sample. How do you advise the clinician in terms of the shipment? How should this sample be shipped? What are some of the temperatures that should be maintained? How should the sample be packaged? Which sample or what kind of sample should be collected? Is it stool? Is it serum? Hmm? Or is it all blood? And if you collect a all blood, are you supposed to aliquot the all blood to get the kind, maybe the right aliquot for the serum? If the aliquot that is actually required is two mils, then you are supposed to get that as a threshold. So if is a stool, how are you supposed to keep this stool so that it's being ferried to the public health? Maybe the public health laboratory is some far. If you are supposed to use a viral transport media, which one would you recommend? If you're used to carry Blair media, which one would you recommend? So those kind of advisory services, we need to offer them to our clinicians so that we get a quality result at the end of it all. Not that the sample has been collected like a spinal cord, a spinal fluid has been collected and is supposed to be shipped for bacterial or viral analysis somewhere else. On reaching the lab, then the sample is rejected, and you know how hard getting a spinal fluid or CSF, cerebrospinal fluid, is hard in the laboratory or in the hospital setup. So let's ensure that we adhere to all the protocols and the advisory part of it we give the clinician. And this one can actually be done during the clinical laboratory interfaces. I tend to believe that we still have the clinical laboratory interfaces in our various facilities. Then effective surveillance required rapid data flow from laboratories to public health authorities. Implementing a laboratory information system allows real-time reporting of confirmed cases of notable diseases, measles or typhoid, to district health officials for timely response. It also strengthens facilities to maintain appropriate safety levels for handling dangerous pathogens. It also prevents laboratory-acquired infections and protecting both staff and the community, which is very much important. Each and every test that you do, or anything that you've got to factor in the issue of the community. Very much important. How is what am I supposed to do so that I also take care or I cater for the community, so the community is not, and even the fellow staffs are not affected by what you're supposed you intend to do. So these ones are just the roles of the laboratory in outbreak investigation. It helps in outbreak detection, that is through testing. It conducts and advises the right specimen to collection equipment, which is very important. The confirmation of the pathogen, it confirms the pathogen. This one is done through genomic sequencing or through culture and sensitivity. It also helps the detection of the new agents. It may be if something is suspected, so it laboratory help us to come up with the new agents. Then identification of mutations of the agents. So you can also, or through genomic sequencing, you can also monitor any mutations. Any form of mutation can actually be monitored through laboratory. Then monitoring of AMR. AMR, I think, is now the new kid on the block in Kenya, and everybody is talking about AMR. So how is laboratory positioned in terms of AMR, antimicrobial resistance, and is monitoring in Kenya? So this one also calls for what we call One Health approach. So whereby One Health approach has got what we call the human, we've got the animal, and we've got the environment. So most of the things that we do as human beings or the healthcare workers, how does it affect the animals, and how does it also interrelate with the environment? So we are advised that we are supposed to be very much privy, and we have all the rest of the cadres on board. So you talk about healthcare workers, we talk about the veterinarians in terms of the animals, then we talk about environmentalists in terms of the environment. So somebody maybe would be wondering, how is AMR affecting me? So the kind of sprays and or the kind of drugs that are used in the animal world or in the environment, the kind of pesticides that we use in our plants, they really affect the human being. How does it affect the human being? The human being eats the chicken, eats the veggies, and also feeds on the animal products. So if we use a lot of chemicals in the environment and the animals, so the end product is the human being that will actually ingest this, and these ones will do. So in terms of AMR, we should also work on that. It also confirms the end of an outbreak. I said this one we can use what we call epidemiological curve, and we plot our cases against time, just against time. We've got the dates and the cases. If you plot it using bar graphs, pie charts, histograms, you are able to do that, and you'll do it well. Then environmental analysis and soil analysis. So basically about the environmental analysis, what are some of the cases that you will actually do environmental analysis? So when you're basically talking about maybe the water analysis, you pick, you are suspecting a cholera case, and maybe the point of origin is maybe a river, you need to pick some of the environmental analysis so that the water is tested for various various organisms. Why, in terms of soil analysis, which organism or in what kind of background or outbreak do we do soil analysis? So soil analysis is usually done when you are suspecting anthrax. So we know how anthrax usually, Bacillus anthracis usually resides in the soil. So the soil analysis is very much important when you're basically talking about Bacillus anthracis. So these ones are just laboratory roles. What I've just talked about, the cholera, the specimen collection, the advisory. How do we advise the clinicians to avoid rejections? So sample collection, I talked about the pre-analytical and the post-analytical, which are very much important. We need to work on that. We advise whether it's stool, blood, stool, swab, in talked of a rectal swab in cases of cholera. How is that supposed to be done? How are they supposed to be kept? When you talk about blood, what kind of volume are we supposed to collect? When you talk about stool, what is the size of the stool? So those kind of advisory services are very much important. As laboratory officers, we need to be privy to those ones so that we advise the clinicians and we get the right samples for analysis. So confirmation of the pathogen, detection of the new agents, I talked about that through PCR. We've got identification of new mutants, monitoring of AMR, which is very much important when you want to talk about the resistant genes or the gene markers. very much important. You want to talk about the phenotyping or microphaging of typing of the organisms. Very much important. Sensitivity, drug sensitivity to come up with the resistant organisms. Very much important. And this one is done in the laboratory. There's no any other place that this one is done. So this one is purely done in the laboratory and with the laboratory personnel. So confirmation of the end of the outbreak, I talked about this, which is very much important. We plot the cases that we've got vis-a-vis what the positive cases that we've got and the number of days that we've got, so that we monitor. Is it plateauing, or is it at the peak, or is at the decline phase? Then special molecular cases. We've also got special labs that are able to do molecular assays apart from our routine laboratories. Then they are also able to do gene phenotyping. They are also able to do virulent factors confirmation and mutant sequencing, which are also very much important. Then laboratory environmental, I talked about when you talk about water analysis, in terms when you are suspecting wash related, that is water related and hygiene related outbreaks. When you talk about soil, you are basically talking about bacillus anthracis in case of anthrax. Then confirmation of the disease of the marked elimination. So we've got also diseases that are marked for elimination in Kenya. The diseases that had been eliminated, like the guinea worms, that is Dracunculus. Then how is the laboratory helping us in elimination of this? Just through testing and monitoring of these cases. So these are the priority diseases or pathogens that I talked about. We have the viral respiratory infections. I talked about that. Which is very important. Vaccine preventable diseases, measles, polio, yellow fever, and rubella, which is very much important. We have bacterial diseases, meningitis, that is Neisseria meningitis, strep, pneumo, anthrax, cholera, and plague. It's usually done through culture, PCR, and AMR resistance testing. These are the laboratory diagnosis. Then we have the arboviruses. We've got dengue, we've got chikungunya, Zika, Rift Valley fever that I talked about that causes abortion mostly in animals. So if you come across maybe a certain area is experiencing, animals are really aborting, and people are also presenting with fever. People are just having fever. This that kind of relation. So you actually have to think along that line. And this one is usually confirmed using special test. We are basically looking for the immunoglobulins, maybe from your immunology. Those days, you can actually maybe factor in those ones. You're supposed to test in Rift Valley, you're supposed to confirm using what we call immunoglobulins. So you are supposed to test the IgG and the IgM. So maybe to refresh your mind a bit, IgG usually clears very fast in the system. While IgM usually takes a longer period to clear in the system. So if you test it and you get IgM, it would tell you that this one is a recent infection. While if you test and you get IgG, it will tell you this one was maybe an old infection and that kind. So you're supposed to do the IgG and the IgM just to confirm the globulins, to confirm the RVF, which is very much important. The same as dengue, we also got leishmania, which is usually also confirmed just using the kits. But though the kits are usually very much expensive. Then we've got malaria. I talked about malaria in length, which is parasite detection and molecular state. Then we've got of late, the country has been experiencing many cases of monkeypox, that is mpox. So as laboratory, we should also be very much aware on how the mpox cases are supposed to be collected? How are they supposed to be shipped? How can we follow up this kind of cases from the index cases? So we've got the index case is made the first person. How can we follow up on the contacts so that we nail down on this? So we say maybe from the index case, this one actually developed this kind of this, and these are the contacts of this. So the contact tracing is very important. It's usually done in majority of the cases, more so in outbreak investigation. So just to wrap it, laboratory diagnostic needs for priority diseases, multiplex PCR for respiratory or airborne panels. We've got rapid RDTs for field use on outbreak setups, though confirmation using MRDT. RDTs are supposed to give you a preliminary, but you've got to confirm it using culture, and it may be also PCR. So we've also got high resolution, then serological testing for vaccine preventable diseases that I talked about, the measles, the polio, and the rest. Then antimicrobial susceptibility testing, especially for bacterial meningitis and TB. AMR. So how are we positioned? How are we? What is the status of us in the laboratory when we come to simple detection and monitoring and detection of the outbreaks in our various facilities and our various laboratories across Kenya? So I'll pose that question to you. So key benefits, proactive problem detection. It also gives us improved efficiency. It also has what we call enhanced credibility, professional development, and better patient outcomes. Very much important. If you are able to do this clearly, we are able to contain the outbreaks, we are able to do proper surveillance system, then we would be home and dry. So take-home messages, laboratory surveillance and outbreak monitoring systems are very important when functioning effectively. They also represent one of the highest return investment in public health, preventing the disease. So our goal is prevention of the disease and is the prevention of the cases. Once you detect a case, then you're able to control it. That is the essence of it. While surveillance is just a continuous process, you would be doing is a continuous process of collecting, coalescing, analysis of all those kind of events. Then we shall actually have saved lives, and we shall also have reduced the healthcare cost through that. Then we've got the strengthening of national and global health security, because maybe just to take you back, how COVID actually ravaged the whole world, and it brought down all the systems to an halt. So if you are able to control a disease like we've got several Ebola cases, outbreak cases in DRC Congo. If you've got the capacity and we're able to do that, if you are called upon as laboratory officers, then I think we'd be home and dry and we'll do it well. So these ones are at the tail end of it. I brought this so that to actually just to tell us how do we, after monitoring, we've done the surveillance, we've got our data, we've got our various variables. How do we plot them? So how do we plot? The first picture shows a trend, a line graph. So this one usually help us in coming up with a trend over time. So we are able to X and Y axis. We are able to plot the cases so that we monitor the trend. So the first one is how do we monitor the trend so that we see what are the increases, were there some kind of upsurges, is there a decline in terms of cases. Then the second one, I talked about the variables, which is age and sex, which is very much important. In terms of age group, which age group is the most at risk, which age group is mostly affected, so that we do what we call target interventions. I think that is home and dry, and you've got to label them well. This one shows the same, how we can come up with, this one was a proficiency testing for laboratory staff in another facility somewhere in Northeastern. What was the performance of the TPT, the proficiency testing when it comes to malaria, which is very much important for our laboratories. What is the performance? Because if you cannot get it well in the blinded testing of these PTs, then we are not getting the result. So the people, our patients and who are our customers are not getting the real results. So this one was just basically a graph showing the PT performance in terms of malaria. That is in terms of detection, speciation, and quantification, which are very much important when somebody would want to talk about outbreak investigation and our routine day-to-day activities. So these are some of the species that were gotten. This one is a table summarizing an outbreak. What were the cases in Plasmodium falciparum, Vivax, Ovale, and unidentified? So this one is also a graph showing how a DQA, that is data quality assessment, was done in one of the facility. In terms of epidemiological weeks, I talked about the EPI weeks. So this one was in EPI week 20, 21, 22, 23, and 24. Then of much importance is the epidemiological week, that the epidemiological curve that I talked about. So this one shows the number of days and the number of cases that we've got. So we've got the number of cases and we've got the EPI weeks. So this one was plotted against the EPI weeks. So you can actually see the outbreak started sometimes in EPI week 14. It went ahead. This one was a continuous outbreak. It went ahead, and in week 23rd, we had the peak of the infection. Then it came down, it declined. Then week 27, we also experienced another peak. Then from there, there was a steady decline until week 31. So having this, I've said, is something that is very simple. You go to your laboratory register, you check on the dates, and you check on the positive cases that you've got. Then from there, you go to your computer and you plot the cases, y-axis and x-axis. So you plot number of cases vis-a-vis the weeks that you've got or the days that you've got. After that, you will be home and dry. You shall have actually monitored the cases, and you can also tell there was an uprise, there was an upsurge here, and we managed to contain it. What was actually maybe the reason, or is it still on, so that you put more interventions on the same. Then this one is also just a table showing data quality assessment that was done sometimes back. Then this one is also in terms of category of cases. That is why I talked about as age being a very important factor and residence being a very important factor. So you group them in terms of age groups, you'll come up with the most affected age group. Very much important. So at the end of it all, I think I'm on time. I'd like to acknowledge Lab Medicine Society of Kenya for giving me the opportunity to present and to share with our fellow laboratory officers the importance of strengthening laboratory processes in our places. That is through quality monitoring systems and strengthening and upscaling our tests in our various facilities. I also want to take this opportunity to thank the Field Epidemiology and Training Program where I am. Then I'm placed at National Malaria Control Program. I want to thank them very much. and above all, the county government of Kisumu, who are my employer. Then Ministry of Health, which is the mother of all the activities, mother of all everything. So I think by that, I think that one marks the end of our presentation. Back to you and over, Bruce.
Thank you so much, Duncan. That was a well-researched presentation. It was very wonderful, and we appreciate you for that. I can see from the comments, I can see Celestine Mulwa saying good presentation. Fred Otawa, that was a good presentation. Fred Njuguna, good presentation, sir. This is so nice, sir. From Victor Umondi. Sami Wanyonyi, good presentation. Mercy Lufoka, good presentation. So generally, it was a very good presentation. We appreciate your time and for taking us through disease surveillance, outbreak monitoring, and early detection. I'm asking the attendees, I also want to thank you so much for taking your time and staying within this presentation till the end. If you have any question, you can post it in the Q&A so that our speaker, Dan, can take us through it. So very fast, Dan, I can see a question from the chat box, a question from Lillian Ouma. Lillian is asking, how do we collect measles samples? Over to you, Dan.
Thank you so much, Lillian, for that wonderful, question. So over time, in my practice as a laboratory officer, we used to collect measles samples from patients, suspected patient children, mostly back then, and we used to collect samples in a red top. Then we used to aliquot. You just centrifuge, because what is needed for analysis is serum. So after you've collected the sample in a red top, because we need a serum, then you aliquot it, you come up with the serum, then you can ship it to National Public Health Laboratories. That is what you used to do before. Thank you, and over, Bruce.
Thank you so much. Another question from Dennis Ilaya. Dennis is asking, it was a good presentation, Daktari. I was one of your students at JOTRH back in 2012. My question is, what is the fate of lab officers now that we play a big role in terms of disease surveillance, yet other cadres are given good positions at the county and national levels? Daktari, you can respond.
Thank you so much, Dennis, for that question. And I think Dennis was my student back then when I used to work in JOTRH. So Dennis, this one is also a wake-up call, just the way I've just mentioned, that 99% of outbreak investigation cannot be declared if laboratory is not there. So laboratory, 99%. Only 1% is for the clinical bit of it. Very much important. So let's, as laboratory officers, kindly let's rise to the occasion. It's only that we generate data, we don't analyze the data. After the generation of the data, then we blame other cadres that they utilize our data for other decision making. Like I would actually maybe want to challenge the officers who've joined. How many of us have managed to analyze the kind of data, the routine data that we've got in place? How many of us are doing line graphs? How many of us are doing graphs? How many of us are doing pie charts? How many of us are monitoring the indicators? Just to start by that. So let's rise to the occasion, because I've talked about the IDSR, that is MOH 545 that we have in our facility. And we do usually report malaria cases in weekly basis. We report from Monday to Sunday. Then we give them to public health officers to report it in KHIS. Have we ever wondered the kind of trend that the kind of results and the kind of trend that we are doing or we've got as a facility? So let's be smart. Let's up our game. The only partition that maybe I like to tell maybe our colleagues is that let's be smart, let's up our game, and let's be vigilant so that we up this. This thing is very simple. Let's claim our position and we walk away with it. Thank you, and over, Bruce.
Thank you for that an eye-opening insight. Maybe you can organize for another session. You will tell us the secrets on what to do with the data for those of us who do the recording and the report to the public health officers. So, Daktari, a question maybe I can ask this question is what measures do you take to ensure that even in the private hospitals or in the private facilities, the data that they have, that they report it faithfully, and they also report it, or let me say, escalate the outbreaks that they get from their facilities to the relevant authorities. How do you make sure that they are faithfully reported? Thank you.
Thank you so much, Bruce, for that wonderful question. Maybe for the privates, because we also just report. I talked about the KHIS. The KHIS is the Kenya Health Information System. I think that is the kind of system that we've got, and the kind of system is interoperable. When you basically talk about interoperable, that is interoperable with what we call laboratory information system. What is gotten in KHIS if you've got the rights. So the only undoing that we've got in most of other facilities is that we don't have the rights to even monitor the data that we generate in our public and private facilities. So as we just report, but we are not able to monitor this. The only people that have got the rights are the disease surveillance and the records officers. So we are not able to monitor this, and we are not able to monitor this. So if you can start with monitoring the indicators, Bruce, I think it will be like you are doing malaria tests. So are you able to monitor the trend by just plotting the weekly basis so that you monitor the trend? How many positive cases were in this month? So in this month, I had like 20 positives, which is unusual against what was gotten. So you should have a mirror. Like in January, how many cases did we report? You're doing a comparison per se. In January last year, that is in 2024, in January, I had 30 malaria positive cases. But in 2025, I'm having 240 cases. You know, that is a serious peak. You can just have your internal, is the buck stops with you. Let's start it small as we escalate it further. But I would really wish for better, for proper and better analysis of this. We usually recommend in most of the facilities that you have what we call facility data quality audits. Facility data quality audits or data, we've got what we call the meetings. So before the data is sent to the highest level, or maybe to the district, or maybe to any other facility, you're supposed to sit down like in a facility and you assess your data so that what is it that your health HRAO, health records officer has that wants to report to the county. Are you able to own up your data? Are you able to own up your data? Well, that is the first step. Are you able to own up the data that you've generated at the laboratory? If it's projected, will you able to own up your data? That is what this is what I did, because we've even seen in other places whereby somebody reports that we had this kind of cases, yet in the laboratory and in your registers that you've got, the hard copies, those cases are not there. So there were some kind of transcription errors through HRIO. So data meetings are very much important. Quality data before the data, or the data is wired or is sent to the other authorities. So I would say meeting is very important so that people discuss the data that generate, whether be it monthly or be it weekly. Very much important, Bruce. Back to you.
Thank you so much. Another question from Wilfred Moriasi. Wilfred is asking, why are these people from public facilities think they give accurate reports, and yet 90% of them are treated in public facilities?
So thank you so much, Bruce, for that. I think is not generally that, and I think if you can get we can have like data meetings, then we can work on this kind of discrepancies. The other day I talked about, my last presentation was about data discrepancies. It was about generally about data concurrence. So when you basically talk about data concurrence, you basically talk about data completeness, data accuracy, timeliness, and completeness. Completeness, data accuracy, timeliness, and verification factor. So how do you get the verification factor? You would say this data is not right before you do what you call the verification factor. Verification factor is usually gotten from all those what I've talked about. You've got to compare the kind of the source documents with what we call KHIS. So what was it that was keyed in the KHIS, and what was it that was keyed in the source documents? So just to give an example of malaria, I usually love maybe giving an example of malaria. So in terms of data, I want to calculate the data accuracy or data verification in malaria. What are the source documents of malaria apart from the laboratory register that we've got, MOH 240? MOH 240 is the laboratory register, but where does it originate from? Where do we get the suspected cases, tested cases, and confirmed cases? We get them from MOH 204A and B, which is outpatient register. We get them from MOH 705A and B, which is outpatient summary register. Then we get them from KHIS. What is reported in the KHIS? So you've got like 4 documents to verify. What was recorded in the lab? What was recorded in the OPD? What was recorded in the KHIS? So that you calculate the 3, then you come up with what we call data verification factor. So that you say there was a serious underreporting and there was a serious overreporting. So any value that is the standard dictates that any value after calculating the verification factor and you get a value that is or less than 0.9 or is less than 90, that is overreporting. If it's less than 90, that is overreporting. If you get a value that is more than 1.1 or over 110, then you say that is underreporting. That is under data accuracy, which is very much important. Thank you, and back to you, please.
Thank you so much, Daktari. At this point, I want to say we have come to the end of this webinar, and I want to assure all the attendees that they'll receive their points. Those are their CPD's points and their certificate of attendance within 24 hours from now. And Even to our speaker, we thank you for taking the opportunity to research and to present this wonderful topic with us. And I just want to take this opportunity to just give you a moment to give us your closing remarks before we end the webinar.
Thank you so much, Bruce, for that wonderful moderation. This one is just a parting shot to our fellow laboratory officers. Kindly let's rise to the occasion and claim the lost glory. We are generating a lot of data as laboratory officers, and we are doing a lot. Just have a look at the kind of the importance that lab plays in outbreak investigation. Have a look at it. Majority of the diseases, if it's not lab, then who will confirm them? Who can pronounce this? So let's up our game. Let's be vigilant. Let's learn. Let's start from. Let's start from. Let's start small as we upscale. I think last time I gave a lecture on descriptive analysis, which was very much important. How do we analyze our data? I did some lecture on analyzing laboratory data to inform action. How do we tell them that this is what we do if we cannot analyze our data? We can always start small, then we come up with something we upscale as time goes by. In laboratory, the data that we also generate, we're supposed to come up with the abstracts, the manuscripts, and publications from those data that we generate after the analysis, so that you speak to the world. Let's speak to the world as laboratory officers. Let's speak to the world. If you do something and you don't speak or you don't communicate, nobody will recognize you. So that is the reason. Let's analyze what we've got. Let's do our test. Let's put them into scientific papers. Let's have abstracts. Let's have manuscripts. Let's publish them, and let's make our profession better. Thank you, and over, Bruce, for the opportunity.
Thank you so much, Daktari. Let us communicate to the world about what we do. Thank you, and for the attendees, thank you for your time, and let us see each other on Monday from 3 p.m. Goodbye and have a wonderful evening.
Goodbye.